Hello everybody and welcome back to the lectures. So, in the previous lecture in this module we are basically talking about modified oligonucleotides, how we can develop new chemically synthesized oligonucleotides that are different than DNA, that are different than RNA. There are many numbers of modified oligonucleotides that have been synthesized and that are available in literatures. Out of those I am talking about mostly two of the modified oligonucleotides which are of major use in biochemistry or in organic chemistry as well as bioorganic chemistry. So, one of them that I have talked about is PNA, peptide nucleic acids, that is different than DNA in the sense that it has a peptide backbone instead of phosphates and sugar backbone. So, normal DNA has a sugar phosphate backbone, PNA has a peptide backbone, peptide backbone. Both of them has nucleic acids. So, PNA also has the nucleobases attached. So, that is the structure of the PNA, and its advantage is that we have talked. First advantage and the most important advantage is that it has a stronger binding affinity towards its complementary DNA or its complementary oligonucleotide. It can be DNA, it can be RNA, it can be even another PNA. And we have also shown that if you have a single base mismatch in a large target DNA, then PNA DNA double helix, double strand melting temperature would be changed, or there would be a difference of melting temperature by around at least 20 degree due to presence of a single base mismatch. So, PNA DNA binding is pretty strong, melting temperature is very high, 73 degrees Celsius. The same double strand, the naturally occurring DNA has a melting temperature only 23 degrees Celsius, and comparative PNA DNA will have very high 73 degrees Celsius. So, binding or the hybridization, degree of hybridization or the strength of the hybridization is pretty high for PNA DNA double helix, and that is the use of it, that you can use this binding property or this hybridization property to find out a presence of single point mutations. And we have talked about that these are some of the patterns, the binding patterns that are generated because of PNA. This is called DNA double helix. This is DNA double helix, and if you have a synthesized PNA which is complementary to part of the DNA, maybe this, then this PNA has the ability to disrupt the local hybridization of the DNA and makes a new DNA PNA hybridized complex or double strand, because this has higher energy. So, this is called the displacement of the DNA double helix. Another one is simply if you have a single stranded DNA and if you have a PNA, that they will hybridized. Today I will show that how we have used, or other people also have used in a different manner, PNA, use of PNA. How can you detect presence of a single base mutation? So, in this case I will talk about first the relevance of finding out the mutations. Why do you need to detect the presence of a mutations? Of course, all of the tumor cells have one or more than mutations present in their diseased gene. So, apart from that, this are from the agricultural stuffs, that the pathogens that attack the agriculture products, crops. Cucumber downy mildew is a kind of pathogen that will damage the cucumber leaf. You can see this is how it looks like when the leaves are attacked by the pathogens. Rice blast is another kind of pathogen that kills rice. Wheat powdery mildew, also you can see the white spots on the leaves that are affected by the pathogens. Corynespora leaf spot, the same thing, the leaves are damaged and the crops are killed basically by the pathogens. So, mostly when the crops are attacked by pathogens or other insecticides, then we use pesticides to kill those insecticides, right. So, strobilurine is a fungicide. So, these pathogens are basically the fungus. So, this cucumber downy mildew is a fungus, rice blast is a fungus, wheat powdery mildew is a fungus, and this also is a fungus. So, you use fungicide to kill those fungus. So, strobilurine is fungicide that is usually used on these kinds of crops. So, this is the structure of a variation of strobilurine. This is azoxystrobin. This is the structure. How does it work? Is a fungicide that are used as agricultural chemicals in many countries, because they have a broad spectrum of control against a large number of pathogens on various crops. So, this is a widely used fungicide that can kill lot of pathogens and lot of fungal attack on the crops. How does it work? It binds to the subunit protein of cytochrome BC1 complex. So, cytochrome BC1 is a part of the protein, and that part, that is basically the active site of the protein, and that active site binds, or the pocket of the active site binds with this product or with this organic molecule. And as a result of the electron transport chain located in the inner mitochondrial membrane, thereby inhibiting the fungal respiration. So, once this molecule binds or docks into the protein, then the protein cannot do its function, and that protein is associated with the fungal respiration. So, if the protein cannot function, fungal growth will be stopped and the fungus should be killed eventually. So, that is how this molecule works. So, over the time the pathogens are clever, and it happens not only for the crops pathogens, but also for our different types of antibodies we develop, different types of antibiotics we take, we consume, and the virus or the fungus, they actually mutate themselves, they become immune to those antibiotics. So, if you remember, penicillin was one of the most widely used antibiotic, and now the microorganisms, they have developed immune system against the penicillin. So, we have to use most of the times different kinds of antibiotics apart from penicillin. Penicillin group antibiotics, but not exactly penicillin. So, different molecules basically having the same property as penicillin. So, because the microorganisms, they can be immune, or they can develop, they can change their genomic systems very fast once they see that they are under threat. So, the same thing happens for these kinds of fungus or this type of pathogens also for the crops. These crops, they mutate themselves in their genome. So, this is the sequence of the wild type DNA that is present in the pathogen or in the fungi. This is the most important part of it. So, G. So, this is the parent DNA. So, GGT, and this actually codes for the amino acid glycine. So, that is what is the function of this wild type DNA, that this part of this codon of the wild type DNA, it codes for the amino acid glycine, and because of this, so it is this active site is tryptophan, glycine, alanine, threonine, and that has the structure of the protein that constitutes a structure of the protein, 3-dimensional structure. It makes the cavity or the active site of the protein, where that strobilurine molecule comes and docks into it perfectly, and that is how the pathogen gets killed. Now, because of excessive use of the fungicides or those organic molecule strobilurine, the microorganisms or the fungus have mutated itself. How? This G, they have mutated into C, cytosine. So, this has now become a GCT. Now, what does that do? The single change of this mutation actually changes the amino acid. It now codes for alanine instead of glycine. The moment it becomes alanine, rest are all as it is, the active site of the protein gets changed. So, the organic molecule, this which was bound initially, which used to be bound to the active site of the protein, can no longer bind, and therefore, it cannot kill the pathogen or become resistant. So, the microorganisms become resistant to the fungicide. So, this mutation, this single point mutation actually saves the microorganism from getting killed. So, they will persist and they will destroy more crops. That is how they have become resistant. Similarly, downy mildew have the same mutation here, same change of amino acid. Eggplant downy mildew have the same, even rice blast has the same, exactly the same mutation and exactly the same change of amino acid, and that is how the microorganisms are now resistant. So, we have developed actually, so this is a long DNA, around 60 mer DNA. So, question is, how do you find out the pathogens, whether the pathogens are mutated or not? This is a single point mutation out of 60 long base DNA base pairs. How do you find a presence of a single mutation, single change? So, exactly same thing happens for the disease when tumor cells or cancer cells is mutated. How would you find out the presence of a single point mutation? And that we have done using PNA as a molecular probe. So, this is the structure of PNA, this is the structure of DNA I have talked about already. Advantage of using the PNA, I think I have mentioned already, the PNA is neutral backbone, there is no charge in the backbone, and therefore, it is an uncharged species. PNA hybridization is independent of ionic strength. This is very important actually. Since this is neutral, no matter how much salt you use or you do not need to use a salt, when you have a PNA hybridization, you do not need to counterize the charge. Usually you have to use sodium or magnesium chloride when you have DNA hybridization to neutralize the negative charge on the phosphates. That is not required in PNA. PNA probes are better target specific and has higher melting temperature than its DNA analogues, that already have been discussed. So, now what we have done is, I will show you a change of modifications, that we have a PNA and that we have attached with a polymer which is a heavier weight. So, it will make the whole thing bulky. And now, if you have the wild type DNA and the single mutated DNA, just we have taken this DNA, this is the wild type with the parent genomic sequence and then the mutated with the single point mutation sequence. Then what happens? The wild type DNA is perfectly complementary to the PNA that we have used. So, they will have a full hybridization, proper hybridization, and the mutant will have weak hybridization or almost no hybridization here. So, that will make this whole complex bulky, and this here your mutant DNA will move. So, this kind of video or this is animation I had shown you before also. We have used this for actually for the PNA and the polymer analog. We have done capillary electrophoresis, affinity capillary electrophoresis that I had talked about before. This is our PNA and this is the polymer, bulky polymer, which is polyethylene glycol that was attached to the PNA, and this PNA is complementary to the wild type, which means it has one base mismatch with the mutant, and this is a scrambled sequence. Basically, that means that has no similarity, that does not form any hybridization with the PNA, and this is injected into the capillary. and now this is your sample. We have injected a mixture of the wild type, mutant end and a scrambled DNA which does not hybridize at all with the PNA. Now, what it does? It will go. So, once this moves through the capillary, the wild type, it should hybridize here actually. I have done. So, wild type and the PNA, they forms a strong complex. Mutant, because of its single point mutation, it changes the melting temperature by around 20-25 degree. So, under a experimental condition, if you do this experiment with a little higher temperature, let us say 50 degree, then this hybridization would be very weak. They will hybridize and open up, hybridize and open up, because it will be in the melting temperature range. So, therefore, because of the strong hybridization, it will move as a complex and you have bulky polymer. So, it will move very slowly. Mutant will have very weak hybridization. So, it will move faster than this, and scramble has no hybridization. So, no interaction with your PNA, it will move straight, and that is what it will look like. Scramble will be something here. I will show you the actual data. Then will come the mutant, then will come the wild type over time. So, by this method you can actually separate out the presence of a mutation, that whether you have a mutation present in it, number one. Number two, how much amount, and this quantitative detection of mutations is very rare actually. There are very limited number of methods that are available which can actually detect the number of mutations. The qualitative mutations can be detected, quantitatively it is actually hard to do. So, this is the actual result. This is the wild type, this is the scrambled sequence. As you move on different parameters, this is the wild type, this is the mutant, this is the scramble. So, mutant and scramble are appearing very close together, wild type is far apart. So, well separated here. So, in this case what we have done is, this was our PNA that had the polymer, right, and this one was the target DNA, wild type DNA, and that was forming perfect complementary here, complex formation. Now, instead of this PNA polymer, if you use the DNA, same thing, DNA polymer, the exactly same sequence, but in the DNA, and if you have the same wild type target DNA, this is where I am trying to show you the important the advantage of PNA. Then this will have a double helix, PNA DNA double helix, DNA DNA double helix. If you use this and do the electrophoresis, see, mutant and wild type, they are not separated, they are merged together in this peak, which means the presence of a single base mismatch in the target DNA is not been able to be detected by using the DNA as the probe, because the hybridization between the perfect complementary and mutated, both are almost similar. Melting temperature is not very different. So, thermodynamic parameters are almost the same. That is why they are both of them are moving as a complex almost similarly. That is why you are not able to detect them. PNA on the other hand is able to detect all three of them: scramble sequence, mutated sequence, wild type sequence. Very nice well separation. This is a beautiful separation actually, very clean separation that you can see. So, because the binding affinity of the PNA with the target DNA is high and with the mutant is very low. So, that is the distinction. So, one of the example, there are plenty of examples using the PNA and using LNA, using the other oligonucleotides to show that their target affinity are pretty high and pretty selective actually. That is what you mostly want, pretty selective. So, this is the PNA structure, this is the LNA structures. So, I have so far explained the importance or the advantages of using the why the oligonucleotides, why the modified oligonucleotides are synthesized in the laboratory, and a lot of people are doing a lot of research to develop new kinds of structures which will have importance in detecting chemical processes or biological processes in the living cells, and I have talked only two of them. So, now I will be talking about the synthesis of it. How can you synthesize these kinds of oligonucleotides? So, I will start first with the PNA. So, again we have to look at the structure of the PNA. This has the backbone which has the ethylene diene CH2, CH2, NH2 and N. This N is attached with basically a amide bond, and then you have the nucleobase attached to it covalently. Then here you have CH2 intervening. So, this is basically kind of a glycine. So, CH2 N and then CO, and then that is fused with the first amide bond, first peptide bond is synthesized here, and then it is going on like this. So, basically here to here is your monomer. This is your monomer, and then they are fused together different with the different nucleobases, and you can do the peptide coupling. So, all the peptide couplings are happened. So, basically all the monomers are attached together, and this peptide you are synthesizing the peptide bond. So, you will be using the same chemistry as peptide coupling that we have done before, and that is the difference with the DNA. So, in DNA you had a phosphate, now you have a peptide, you had a sugar, now you had this, and then later we will see once the PNA is finished, then I will talk about the LNA which has this structure. So, if I write the monomer CH2, CH2, N, CO2R basically. Here you can couple, you can attach the nucleobase CH2, CH2, NH, BOC. This would be your skeleton, backbone skeleton, and on top you will attach the nucleobase here, and then you have to make this into an acid, then you can go ahead with the coupling. So, how do you synthesize this? First is CH2, CH2, N, NH2, NH2. We start with this. This molecule is ethylene diamine. So, what you have to do is you have to make one of these NH2 react with this, and the other amine you have to protect by the BOC. So, first is the protection by the BOC. BOC anhydride if you use, and this is little bit tricky reaction, you have to use one equivalent of it and in a controlled way, then one of this amine would be protected by the BOC. It's a little bit tricky reactions, you have to be very careful, so that the both are not, usually if you use the BOC anhydride or any other electrophile, both of these NH amine will be protected or will react. So, you have to be very careful, so that you can get only one side protection. Next reaction is this with the, you have to make a CH2 and a S star, right. So, this is done CO, let us say ET we take here, CH2 Br. Now it is easy. I am not writing the exact the other reagents, I am writing the main component. Substitution, you will have NH, CO2Et, and you have NH BOC. Okay. Then your one skeleton is ready. Now you have to attach this with the nucleobase. If you take thymine, this is your thymine here, treat this with Br CH2 CO2Et in presence of mild base potassium carbonate, that is one. Then what will happen? You will have the same chemistry that we have seen, nucleophilic displacement. So, you will form is amine this, and now if you hydrolyze with sodium hydroxide, sodium hydroxide would hydrolyze this into COO minus, sodium salt of the acid basically, and then further you have to neutralize it into an acid. So, you have to use HCl, because after hydrolysis what you will have? You will have NO COO minus Na plus, and that you acidify using the HCl. So, it will be COOH, that will give you this. Of course, there is still a chance that when this is reacting this NH can also react, that can also happen. So, under suitable experimental conditions you have to optimize the reaction condition so that the reaction of the side products is low. So, this is done. If you have cytosine, NH, then you have to, if you just treat this with this reagent, then nucleophilic reaction will happen here. At the same time, even a better nucleophilic reaction would happen through the amine, this side residue amine, and that you do not want. So, you have to first block this, protect this. pH CH2O CO Cl in presence of pyridine, you need a Lewis base. Then what you will have? You will have in NH COO CH2 pH. This is a protection of the amine, and now you can react this with CH2 CO2 ET and get your selected H and then this is 1, number 2 is hydrolysis. So, this will become your acid. You will have this for the cytosine. These are the pyrimidine bases. If you go for the purine base, yes, of course, you have to link it at the N9 position here. So, first thing again you can use the pH CH2O COCl pyridine to protect the amine. N COO CH2 pH. Here is N, and then similarly Br CH2 CEET, number 1, number 2 hydrolysis, that will give you the monomer for the nucleobase. N9 you want. NH, I am writing P for the protection. This is for adenine. Now, for guanine it can be little bit different than a straightforward. You can still do it straightforward way, straightforward protection, and then using this step, or you can start with a modified one. Instead of the guanine itself, you can start with the chloro version of it, because it will hydrolyze and produce a double bond over here. So, the first step is BrCH2 COOET. You can do this protection before also and after also. So, it is better to do this protection before. NH, start with this protection, and then this, it will give you COOH followed by hydrolysis. You have Cl, nitrogen, NHP protection, and you have N here, and then pH CH2 ONA. So, sodium salt of the alcohol, pH CH2O minus basically is anion. If you do a straightforward hydrolysis, you will have a double bond over here. So, this would be a protected alcohol, even better. Instead of double bond O, you can have the protections, and that will facilitate your further reaction steps actually. So, these are the nucleobase skeleton that you can synthesize. So, I will take one of these examples, for example, with the thymine. This is the one you have synthesized. COOH, you have synthesized this, and plus you have before synthesized CH2, this one was NH BOC. So, on NH and CH2 COOH that you had synthesized. Now, these two can be coupled. So, you have a acid here, you have the amine here, secondary amine. Then if you couple together using the peptide coupling agent, same as peptide coupling that you use, you can use HBTU or anything else or any other kinds of peptide coupling agent. Then you will form a new peptide bond between this acid and this amine. So, this amine CO2ET NH BOC N CO, and here you will have the thymine N. So, NH this. So, your monomer is ready. Only one step is left over. Here you have to hydrolyze the ester into the acid, hydrolysis, as you have done by sodium hydroxide and then followed by addition of HCl to acidify. So, this is your N, that will make you here acid. This will keep your BOC. Now, here there is a CH2, I am sorry, there is a CH2 here, there is a CH2. So, base, this would be your monomer. Now, you take multiple of the monomers one by one and go on coupling them according to your sequence, and you can synthesize your PNA likewise. Now, synthesis of PNA is little bit tricky. Yield is not very high and many steps actually, takes little bit time, and if you want to purchase it, you can also purchase it from some of the pharmaceutical companies. It's very expensive. So, you can try to synthesize it in your own lab if you want to use them. So, that is the organic synthesis part of the PNA, and now I will show you how you can synthesize the LNA. So, draw the structure of LNA. Let us take the example of thymine. So, here there are many varieties. One is of course the one I had shown you here, this starting with the 4' end, or the one I have here is little bit different, that is this OH, and then here is the lock, this is the lock. So, this is another variation of the locked nucleic acids. Instead of here, it can be here as well. Of course, the original one was this, and that was developed by if I can remember correctly, Jasper Bengel is the person who first reported or had shown the importance of the locked nucleic acids. So, very quickly I will show you the synthesis of it. You can start it from here. This you have done before, protection of these two with the acetyl group, and here you start with the ketone and O. You can start with the protection, for example O TMS or TBDMS, anything, maybe a different one. Let us start with ODMT. So, you can synthesize this of course, because DMT protection can be done. This is simple acetyl protection that we have seen in the sugar chemistry when we have done. Here to this. O benzyl, and then you can have the double bond here. This can be ODMT, or here they have done OBN, O benzyl protection for both. So, this is done by, of course, it is a nucleophilic substitution with a vinyl anion, right. So, benzyl bromide done, and then acetylation thymine. So, first step is BU3NF, it's a fluorating agent, and then you have BNBr, benzyl bromide will make it OBN, and then you can have the vinyl magnesium bromide. Vinyl magnesium bromide, basically it is basically a Grignard reagent. How a Grignard reagent reacts on a carbonyl carbon, it will have this is the positive part, this is the negative part. So, this will go and attack here, it will open up the O minus. In order to stabilize the O minus, you can use the BNBr. So, it can take OBN here, that is how it will be forming. And now what you have to do, if this is your target, you need one CH2, here you have 2 CH2. So, you need one CH2 basically here and one CH2 OH, that is what you need in this position. So, you have to hydrolyze it into, or you have to reduce it into the CH2OH. What is the way to do it? If you do this iodine reactions here, sodium hydroxide, acetyl chloride, NaIO4, sodium periodide, NaIO4 treatment will give you, followed by hydrolysis of course. If you remember, if you have a ethylene group CH2CH2, if you treat it with NaIO4, then what you get? You basically get an aldehyde, CHO. That is what you will have here. You have OBN, here you can have OBN, and then you will have the aldehyde here, and then this aldehyde is being reduced to the alcohol NaBH4, sodium borohydride reduction you can do, or you can use the other suitable maybe dibolol can also be used to reduce one electron reduction. This OBN, and that will give you OH here, OBN, and the last step is basically a coupling here. So, we have this. OH protected, here OBN protected. First I will unlock this. Oh, before that, if I unlock this, and there will be one reaction here, either this has to react here or this OH has to react here and open up the eliminate one hydroxyl. elimination of 1 hydroxyl is required here, and if you want to eliminate that, this is not a good leaving group. So, tosyl chloride I will make it a good leaving group first and then we will eliminate. This becomes O tosylate. O benzaldehyde, and then I will open this up, deprotection, OTS, that will give you OH here, it will give you OH there, OBN, OBN, and now if you use sodium hydride is a H minus, basically that will take off this, it will make a O minus here, and this O minus would be very reactive, it can do this reaction, elimination. OH. So, basically now you have O and you have this bond, OBN, OBN, and then this can be hydrolyzed, deprotected to give you OH, CH2OH, here you will have the OH and here you will have OH, and you can do the normal way that we have done the TMS the thymine coupling. You can use thymine, or before the protection, here you can use the thymine actually. I will go here straight thymine T, here you can have the thymine and then TMS that will make the thymine active, and you can do the coupling here, that will give you this, followed by the hydrolysis, deprotection. Number 2 is the deprotection of the all the protecting groups. That will roughly give you the monomer, and then you can carry, you can make this DMT as usual, and this make the phosphoamidite bond, making it suitable for the DNA coupling. So, this monomer is a basically DNA framework. The monomer would be the same as we have done the phosphoramidite chemistry for DNA. So, that you can further do. So, this is one of the examples of the locked nucleic acids, which through which you can restrict the conformational flexibility of the oligonucleotide that you will be synthesizing would be restricted here. And all these have the advantages that this is not a not a natural oligonucleotide, it is more of unnatural oligonucleotide, and many a times they are stable inside the living cells, because the cells or the proteins enzymes that are there which actually degrades foreign DNA and the virus particle virus DNA viral DNA and all this. So, those cannot recognize this as a substrate. So, these kinds of molecules, if you take inside the cells, they will survive longer, and therefore, will allow you to do your biological studies. So, with this basically I will complete the module. So, in this module what we have talked about is we have talked about the synthesis of artificial oligonucleotides, which we call as molecular probes. So, in general molecular probes, basically probes in terms of molecules. So, molecular probes are the molecules or the oligonucleotides or the polymeric materials that are used to detect something in the molecular level, and especially to detect something inside the biological cell. The challenges are, if you want to do that, most of the molecules that you inject into the cells would be destroyed immediately. So, therefore, studies that you want to do inside the living cells cannot be done, because all the probe molecules that you are injecting will be immediately destroyed. Cells do not allow most of the foreign particles to enter into the cells. So, that is where especially PNA, the peptide nucleic acids and the locked nucleic acids I have talked here, because these are the kinds of modifications that can survive in the living cells much longer. So, these are two different classes of artificial oligonucleotides that can be synthesized. These oligonucleotides are highly useful to study various probe target interactions. This is the probe. Target can be anything, target can be protein, target can be DNA, as I had shown that detection of single point mutations in DNA, that is one of the aspect. Protein DNA interactions, whether your protein is manipulated or not inside the living cells, that can be studied or analyzed. So, in general, probe target interactions. Most of the applications of the PNA and locked nucleic acids, you will find that they are to study the RNA, the mRNA, especially the messenger RNA, the presence of the messenger RNA or where the messenger RNA is moving, leaving detection or time dependent detections of the movement of the messenger RNAs in the cells are studied a lot using these kinds of artificial oligonucleotides. So, probe target interactions such as detection of single nucleotide polymorphism, or in short SNP, which basically means the presence of single mutations in the genome. Disease diagnosis, many times the diseases which are associated with gene mutations can be studied using this. And gene therapy, that is another very important aspect of it. If you want to silence a gene, if you want to neutralize the function of a gene, then PNA is one of the good molecule to be used. So, your gene will have higher binding affinity, much higher binding affinity with PNA, and when they form a stabilized hybridized complex, your gene cannot go for further DNA replication, because for DNA replications they have to unwind, and once PNA binds there, same goes for the gene silencer RNA also is similar, siRNA we call them. They actually go and bind very strongly or very firmly with your target gene, and therefore, the activity of the gene is further lost, it cannot undergo cell divisions. So, gene therapy, this is called, or gene, even in the diagnostics, the sequence of a gene can also be detected or also be diagnosed using the molecular probes. Why PNA? I have already shown that PNA, due to its neutral backbone, binds strongly with its fully complementary target DNA. So, and in here once again I will mention that PNA duplex double strand has much higher melting temperature or much higher binding energy compared to PNA DNA, which is of course much higher compared to the natural DNA double helix. So, LNA, due to its rigid locked sugar structure, also is highly target selective. So, whenever you are using a probe to detect your target, so of course, the probe has to be highly selective towards the target only. It should not bind to all molecules, it should only bind to the molecule of your interest. So, selectivity is very important, and that is where these two molecules are highly advantageous of. Locked nucleic acid also is very target selective. Both PNA and LNA are more stable, as I was saying, are more stable in living cells compared to a foreign DNA oligo. So, if you send a DNA, foreign DNA, synthesized DNA into the cell, it will immediately be cleaved by different enzymes present in the cell. They are called the nuclease. Nuclease are the enzymes that destroys DNAs or even RNAs. The reason is being virus and bacteria, they will enter into the cells. So, nuclease, if they can figure out that those species have DNA, they can immediately destroy. That is what our immune system is also. It is a part of our immune system. So, if you use DNA oligonucleotide synthesized as a probe, they are not very stable into the cell, they are not going to persist in the cells, would be chopped off. So, you cannot do your studies using these DNA oligos. PNA and LNA. PNA is highly nucleus stable. It is not chopped off by the nucleus, because PNA has the peptide backbone and nucleus cleaves only the phosphodiester bonds. So, PNA is very stable. LNA, because of its locked structures and very unknown structures, it is also quite stable by the nucleus. So, they will persist longer in the living cells. So, to allow you to do your studies. So, these are some of the advantages associated with the synthetic molecular probes, and I have shown you their experimental utilities also. There are many more things that are there in the literature, in the textbooks also, that you can go through. So, with this I will conclude this module. Thank you.